our sagittal view. That's this muscle right here. Okay, so this part of it is up here. And this part of it right here is this portion right here. Okay. So technically, from this muscle, all the organs that go up to the abdominal peritoneum are in the pelvis, and any structure below that are in the perineum. Okay. So this muscle right here would be what muscle? Which one covers the bulb of the penis? Bulbospongiosus. All right, so that's the one you're seeing here. But we can't see the ischial-cavernosus muscle because that's behind or in front of our screen. Now, in our upright models of the penis, you can see these muscles. And you can see the pudendal nerve. So here's a transverse section through the penis. This top structure here is, the whole thing has the fascia wrapped around it. So these two bodies up here would be the corpora cavernosa, and the ventral one would be the corpus spongiosum. Now if you say corpus spongiosum, I'm not going to mark it, but we only have one. So the accurate term is corpus instead of corpora. Okay. So then if we split the penis in half, you can see those same structures with the septi and the urethra within the spongy urethra, the expansion, which is the glans penis. So it helps to have this put together so you can understand the three-dimensional relationships. So this is an anterior view, all right? Here's the posterior view with the vas deferens, seminal vesicles, and this bright, well not bright, but kind of maroon colored structure is the prostate gland. Anybody know the name of the general term for this muscle that forms the floor of the pelvic cavity? The Levator ani. So remember puborectalis and pubococcygeus and so on? That's what this muscle is right here. And it contributes to the sphincter around the rectum. And then over here, this muscle is covering the obturator foramen. Anybody remember what that one was? That's lateral rotation of, covers the obturator foramen, passes out of the sciatic notch and attaches to the greater trochanter. Obturator internus. Okay, so let me get a pelvis out here. We have this model we looked at last semester. These are ligaments that help to close off the sciatic notch, greater sciatic notch and lesser sciatic notch. So here's the obturator foramen. Remember, the only thing that passes through it is the obturator nerve, artery, and vein. But from the inside, a muscle covers that and comes out, attaches to the femur, and that's the obturator internus. So there is a gap created from levator ani to obturator internus. That's actually filled with adipose tissue. But running along the obturator internus here, you can see some colored lines, yellow and red and blue. All right, that's the pudendal nerve. And the pudendal vessels, which we'll talk about when we do cardiovascular. So if you look here, you can see that pudendal nerve running along the obturator internus and then coming out and sending branches to the penis. So that pudendal nerve gives off the superficial and deep perineal nerves that we're not going to worry about. I just want you to be able to identify the pudendal nerve. All right? Now, pudendal nerve get there. I'm not in my other room classroom at 4:30 because I used pipe cleaners for that. Stuck with a glove. Pudendal nerve isn't that large. All right, but if you look at the pelvis, remember the greater sciatic notch and the ventral rami of the sacral nerves come together and exit as the greater sciatic notch, which passes down the posterior aspect of your thigh, and it's formed of the tibial nerve and the common fibular or peroneal nerve. All right? Passing out with that, coming off of the sacral plexus, and again, not this big, is the pudendal nerve. It comes back in over the ischial spine and travels to the perineal region, just like I showed you on that model. So it's going to go out the greater sciatic, come back in through the lesser sciatic by going over the ischial spine, and now it's back in this region, which is our pubic angle, all right? And now it can innervate the external urethral sphincter, the bulbospongiosus muscles, and the ischiocavernosus muscles. Okay? So now if you look at a Netter's diagram of that, it should make a little bit more sense from the anatomical pathway that it passes through. With this model, last semester we looked at the levator ani as it forms the floor of the pelvis. Okay. And then superficial to that, we had this structure filling it in. Now this is a female, so it has a vaginal opening, right? And so we'll look at this on Tuesday, but we still have an ischiocavernosus muscle that's covering the crus. In the female, the bulbospongiosus muscle is split because it has to go around the vagina, but we can still see the external urethral sphincter going horizontally to form that triangle. Okay. All right, so now let's spend some time talking about the physiology of the neural control of all of this. So we're going to turn back. We'll come back to sexual responses, but I want to go back to the hormones first. We skip those. It's kind of like your nervous system. I never can figure out how I want to organize. You let it one way and talk about it a different way. So it's right after the end of the testis. On page 62. You have this diagram in your lecture notes. And I have both of them up here on the board because they're slightly different. Can I erase the board so I just have your pictures of that? Okay. Every time I erase the board, I think I have to do this all over again in 3 hours.
Just for smart work.
Then I do it. Well, I have to show it a little bit at a time so you guys can follow it through. At Sac State, that's first week, it's all lecture, so even in lab, I'm lecturing again. So I give one lecture, like yesterday in the morning, I did epithelial tissue, and in lab, I did the lecture on connective tissue 4 times. So by the end of the day, I'm sitting on the counter talking because I'm tired. So at least here, it's just twice. Yeah, we're back to the physiological control, all right? There's a little bit of a difference here that I want to show you, some of the corrections, but most of these hormones you're already familiar with. You're even already familiar with which ones use the FIT2 pathway and which ones use the adenylyl cyclase and cyclic AMP pathway, all right? So let's just look at the board first before I diagram it. So we start out with our actual master gland, the hypothalamus, and its hormone is abbreviated GnRH. Does anybody know what GnRH stands for? Gonadotropin. So gonad refers to both either ovary or testis. Tropin refers to growth, all right? So the hypothalamus, and we'll kind of cycle this around, is going to release gonadotropin releasing hormone. How is this going to get to the anterior pituitary gland? Through the portal system. Exactly right. Okay, so it's going to move into cap bed 1, travel in the median eminence right here, travel down the portal system through portal veins to the anterior pituitary, and leave the portal leaves blood via cap bed 2 and enter the anterior pituitary. It is, in some organs, we'll use the cyclic AMP, but in the anterior pituitary, its pathway is the FIT2 pathway, all right? So then it causes the anterior pituitary cells, gonadotropes, to make LH and FSH. So both of those, they're named for the effect in the female. So luteinizing doesn't occur in the male, and it doesn't have follicles. The ovary has follicles. So sometimes LH is also known as interstitial stimulating hormone or interstitial cell, but most places they just use the same term, and even though it really has a different function in the male, it's a similar chemical occurrence. So, and it's the LH and FSH go into the testis, and it's going to get there how? What blood vessel is going to carry it to the testis? The testicular artery, okay? And LH has receptors on the interstitial cells, or interstitial cells of Leydig. Those are the ones that are in between the seminiferous tubules. And the FSH binds to the Sertoli cells. So when it says seminiferous tubules here, it's talking about the Sertoli cells or the sustentacular cells. Then the effect of LH on interstitial cells is to convert cholesterol to testosterone. It goes through a series of different forms. And now testosterone is going to go into the blood, because around the interstitial cells we have capillaries. So it's taken up by the blood and distributed throughout the body. But right next to those interstitial cells are the seminiferous tubules. And it's going to influence, their influence at that location is affecting spermatogonia mitosis and the meiotic divisions of the spermatocytes. So we want to have a sufficient amount binding to the Sertoli cells. Right? Now, testosterone is a steroid hormone or a protein? Steroid. So it's going to pass through the plasma membrane of the sustentacular cells and bind to this antigen binding protein that keeps it in high concentration within the cell. FSH is not a steroid hormone. Okay? So its receptors are on the surface of the sustentacular cells, and it works via cyclic AMP, and it causes these cells to make the antigen binding protein as well as to make inhibin. Inhibin is a direct feedback. It binds to these cells and causes it, so it says it has it going to the hypothalamus. This is the correct one that I want you to understand, or the feedback is this one here. This one is nice because it shows the cells, but its primary role is to go directly to the pituitary rather than to go down through the hypothalamus. So testosterone's negative feedback is up to the hypothalamus, and it inhibits GnRH, which lowers FSH and LH, whereas inhibin is back to the anterior pituitary primarily. Okay. So these two red lines need to be flipped. And that's why I have both of these diagrams on the board. So now let's diagram that and go through it again. So we have the hypothalamus. It is going to send GnRH, which is releasing hormones. Some books call them releasing factors, so the terms are interchangeable. Going to send it to the anterior pituitary, the cat-bed 1 to portal vein to cat-bed 2. I'm not going to write that in. Okay. And this is not positive feedback or negative feedback. It's a direct stimulus. It's like turning on a light. Okay? There's no sensor there that goes up or down. The anterior, and this is going to bind. I'll just draw one line stuff. This is going to bind to anterior pituitary cells. So you have the PIP2. So inositol triphosphate and diacylglycerol and protein kinases and calcium ions and all of that type of event. Okay. Then these cells are going to release LH and FSH, luteinizing hormone and follicle stimulating hormone. These will travel to the testis, to interstitial cells. Let me make that a little bit higher for those of you who are in the back of the room. So here, this hormone now uses the cyclic AMP pathway. Pardon?
Is your phone ringing? Did your phone just ring?
Oh, I didn't hear it. I'll turn it off, it won't. Okay, so in the testis, we have the interstitial cells. So in this picture, LH will bind to the interstitial cells via membrane receptor, activating the cyclic AMP second messenger pathway. And FSH will do the same thing, binding to the Sertoli cells. So it's shown here, it's just not showing the pictures. So the interstitial cells, under the influence of the protein kinases activated by cyclic AMP, are going to release testosterone. Which is a cholesterol-derived molecule. And as you can see in this image here, testosterone is going to go through the Sertoli cells to get access to the developing spermatocytes. Okay? make two molecules, inhibin and androgen binding protein. Androgen binding protein works right there at the Sertoli cells, and it binds testosterone and increases the concentration of testosterone at the Sertoli cells. Inhibin is more, its important role requires it to be distributed back into the blood so it can get to the hypothalamus. I'm sorry, so it can get to the anterior pituitary gland. So inhibin so the higher the inhibin, the less LH and FSH is produced, primarily FSH. So that's the negative feedback for that. Whereas testosterone has a negative feedback primarily at the hypothalamus level. So, if somebody is castrated, maybe they're a sexual deviant and they've agreed to that surgery as part of their release from prison, or maybe they have testicular cancer. It's done kind of like a mastectomy to remove the source of the cancer cells. What would happen to their levels of testosterone? They go down. What would happen to their levels of GnRH? They go up. Okay. In menopause, when we talk about female reproductive system, and the eggs and the follicular cells are no longer in the process of producing estrogen, same thing. GnRH goes up. In fact, that is a biological sign of menopause. Because once it goes up to a certain level in the female, it never comes back down again. All right, so if it's been at a certain level and the woman is in, even if she's 32, all right, she will have gone into early menopause. So it'll normally be up and down with the monthly cycle like this, and then at menopause it will go 3 or 4 times higher, and then it just stays there. Okay. Now, cyclically, a woman is not considered to have gone through menopause until she hasn't had a period for a year, for 12 months. But chemically, biochemically, you can look at the levels of GnRH and determine that. Are there any questions on this? Kind of tying in a little bit of new information with what we've talked about before with endocrine components. These are good diagrams to know. They've appeared as the extra credit components in previous lecture exams. So this is just a review of what we talked about. So we have the GnRH. Goes through capsed 1 to the portal veins to capsed 2. Binds to receptors in the anterior pituitary. And activates the phospholipase C. Now in your homework, some of you put the cyclic AMP. So I went and looked it up. And some recent research, so if you just looked up GnRH, cyclic AMP, you would find articles that said yes. Research has shown that GnRH can activate a cyclic AMP pathway. That is involved in the regulation of the frequency and how much, kind of in the cyclic pulsatile component, but in the activation of the pituitary gland making FSH and LH, it uses the PLC. Okay? So in this sequence, causing the cells of the pituitary to synthesize LH and FSH, it uses the phospholipase C pathway that I talked about up here. Okay? So LH goes to the, also known as interstitial cell stimulating hormone, so you might see ICSH and reference as its function as a male. So in the interstitial cells, as I drew on the board, it does cyclic AMP. And so this is what's happening within the interstitial cells. And we'll look at this again in the female, because look what we see as a pathway of cholesterol. We have estradiols and estrogens. So the male produces a small amount of estrogens, not in the interstitial cells, however. All right, so again, this is a little complicated, and we'll repeat it with the female because that's an expansion of the amount that actually occurs. So cholesterol is taken up by the interstitial cells. In the female, and I realize we haven't gotten there yet, all right, but I'm going to kind of keep repeating this to you. In the female, we have the egg. We'll start calling it an oocyte. Maybe I should just do that. It really is an oocyte, it's not an oogonium, because it's reached the primary oocyte stage by the time the female is born. Surrounding the oocyte are follicular cells as the follicle is developing. Remember that homologous list we're going to be working on? Follicular cells are homologous to interstitial cells. I'm sorry, homologous to Sertoli cells. Then around these follicular cells, we see a layer of cells that as a group are known as the theca interna. Those are homologous with interstitial cells. Okay, it makes sense. The follicular cells are right next to the developing oocyte, and Sertoli cells are right next to, surrounding, developing spermatocytes. Outside of those, we have the interstitial cells. So in the female, outside of those, we have the theca interna cells. Guess which group of cells LH binds to?
The theca internis?
Theca interna, okay? So LH is going to bind to theca interna, and FSH is going to bind to follicular cells. We continue that same pattern. So for years, it never made sense to me when I was doing this lecture that if it was the interstitial cells that made testosterone, if we carried that homology, it should be the theca interna cells that made estrogen. Because one was the male hormone and the other was the female hormone. And it just didn't fit. So finally, after delving around and looking up some stuff, it does. It is a parallel. So let's look at this together, and again we'll repeat it when we get to female. So cholesterol is taken up by the interstitial cells in the male, and cholesterol is taken up by the theca interna cells in the female. Under influence of LH in the male, cholesterol is converted to androstenedione, and then further converted to androgens and testosterone. So this is the male sequence. Okay. So can I erase this first half of the board? You guys have that? And I'll leave the other diagram and kind of go through this with you, not drawing the molecular structure, but kind of writing the words. So we have cholesterol, going to form androstenedione, and each of these is regulated by an enzyme, okay? which in the interstitial cells is going to have been activated by protein kinases under cyclic AMP influence. So in the interstitial cell now, this androstenedione is converted to various androgens, which are converted to testosterone. That's why we have that similar appearance, the four-ring structure of cholesterol.